Hi, my name is Carlos Simmerling from Stony Brook University. Today I'm going to tell you how we're using GPU computing to explore vulnerabilities in the SARS-CoV-2 spike glycoprotein. SARS-CoV-2 is a coronavirus, one of 7 coronaviruses that are known to infect humans. Some of them cause mild illnesses, like the common cold. Others cause SARS and MERS, and SARS-CoV-2 is the cause of the COVID-19 pandemic. It's important to ask whether we're ready for the next coronavirus outbreak. And I think the answer is no. We have a lot more to learn about coronaviruses and viruses in general. And today we'll ask whether HPC computing with GPUs can help us be better prepared for the next pandemic. These are experimental images of the SARS-CoV-2 virus, and it's got protrusions from the surface that are called spikes. These are proteins. You can see little zoom ins on the right that are close-up views of these proteins. And these make the virus look like a crown, which is why it's called a coronavirus. They're quite small, only 100 nanometers in diameter. That's 1,000 times smaller than the width of a human hair. We can zoom in to try to get more detail about the virus and how it works, but it's very difficult to see such small objects. And so I'm going to show you a computer model of the virus, and you can see the gray spikes sticking out from the surface. These are proteins. Each spike is made of 3 identical proteins shown in gray, and in the lighter blue color are sugars attached to the proteins. Here I'm going to show you a lot of pictures like this, where each one of these identical proteins is colored in blue, yellow, or red. The lighter colors are the S1 subunits at the top of the spike, and the darker color is the S2 subunit closer to the viral membrane. The job of the spike is to help the virus infect cells. I'm showing you here a series of snapshots of what we think happens as the virus infects the cell, going from left to right. The viral membrane is on the bottom. That's the surface I just showed you in that last slide, and the spike protrudes from the surface. When it gets close to a host cell membrane, such as a human cell that has an ACE2 receptor, which is a target of SARS-CoV-2, then the spike interacts with and binds to the ACE2 receptor. In this instance, it's got both the S1 and S2 subunits, and it's in the pre-fusion state. After it interacts with ACE2, the S2 subunit dissociates from the S1, leaving behind the S2, and that undergoes a series of conformational changes where it releases these long chains that insert themselves into the host cell membrane like a harpoon. Once the virus is attached to the host cell membrane, it pulls the 2 membranes together and allows the virus to deposit its RNA into the host cell. Once the RNA is in the host cell machinery is used to replicate the RNA and proteins that are associated with it and produce new viral particles. Blocking this process is an important aspect of blocking SARS-CoV-2 infection. Now, the spike has been seen experimentally and studied in quite some detail, and we have better pictures of how the spike looks. There are typically 2 different forms of the spike that are seen. One is the closed form here, where the receptor binding domains at the very top that interact with these 2 are pulled in tightly to the spike surface. Other structures that are seen have an open form, where the receptor binding domain has moved away. In this case, one of them is away from the spike. That's the conformation that it can use to bind to an ACE2 receptor, and a fragment of that is seen in this experimental structure in gray. You might ask whether it can bind to ACE2 in the closed form, so we can dock the ACE2 on, and there's a steric clash and overlap, so that means that it must open in order to bind to ACE2. Now, it seems reasonable that if its job is to bind to ACE2, that it would be open all of the time so that it could always bind to ACE2. Why would it close? And the answer to that has to do with your immune system. And so in this structure of the spike here, I'm showing an open form in 10 different experimental structures where antibodies, shown as gray ribbons, are seen to bind to the same receptor binding domain that interacts with ACE2, as you see over here. So when these antibodies bind tightly to the spike, it can no longer bind to ACE2, and it's neutralized in its infectious ability. different antibodies bind to other regions of the spike, and many of them are able to block the spike from carrying out its function. So the receptor binding domain closes in order to hide from the immune system and avoid immune surveillance and immune binding. Now, your immune system is what's responsible for giving you immunity to COVID-19, and that is the job of the COVID-19 vaccines that have been released. This is a major success story for science, and these stimulate your body to produce antibodies to the spike and bind to it when you're exposed to the coronavirus, SARS-CoV-2. So you might ask, if we have vaccines already and they're very effective, which they are, why do we need to keep studying SARS-CoV-2 and other coronaviruses? And the answer is that the virus is still evolving, and you may have seen news stories that talk about different variants of the virus, such as the B.1.1.7 variant that is now becoming common in the United States. And that and other variants typically these days have this E484K mutation that you see in both of these papers. What is an E484K mutation? Well, that means that at position 484 in the spike amino acid sequence of the protein, then the E amino acid, or glutamate, which has a negative charge, has been replaced with a K amino acid, or a lysine, which has a positive charge. So the virus has made a change to its spike protein so that a negative patch on the surface is now a positive patch, and if an antibody was expecting to bind to a negative patch, now can be repelled by the positive patch and have less effectiveness. And so this is why we need to still be worried about the spike and about SARS-CoV-2. The virus is evolving, especially at the surface. The surface is where antibodies bind. They are also large proteins and can only access the outer surface of the spike. And antibodies in general recognize shape and chemical color, meaning things like the charge that I mentioned on the last slide. And so you can imagine an analogy that you might be tasked with recognizing a Honda Civic. And this shows you Honda Civics through different generations. And if you decided to focus simply on the color or the shape of the wheel covers or the bumpers or the hood, that from one generation to the next, you would not be able to recognize it because those things change. They do not influence the function of the car, and so they're readily swapped out. Now, from the point of view of a car, if we said, well, open the hood and disconnect the battery, that would disable all of these cars. And that's because that's a crucial functional part of the car, and the surface color is not. So we need the same information about these viruses and about the spike in particular. What are the functional parts? How does it carry out its job as a machine? And how might we interfere with that in a way that would be very difficult for it to change? And so that's our goal, is to study the spike and understand how it works in enough detail that we can find targets that are difficult for it to change. We do this with a combination of physics and high-performance computing. This is a review that we recently published, Protein Storytelling Through Physics, and we highlight a lot of the success stories from our groups and others that have used these methods to understand molecular biology and disease. One of the examples that we provide is the spike that I'm going to talk about today. We typically start with an experimental structure of the protein, like I showed you before for the spike, and these can be very high quality structures, as the protein you see here, but they're missing information about flexibility and motion, and that's because the experiments average over a long period of time relative to these motions. Most of them also average over many molecules, and so we don't get information about flexibility. With our simulations, we think that we can add this information in about dynamics and flexibility, and as the Nobel Prize winning physicist Richard Feynman said, everything that living things do can be understood in terms of the jiggling and wiggling of atoms. And so that's the goal of our simulations, is to understand flexibility and mechanics. How does this work? These are called molecular dynamics, or MD simulations. And here's a protein that I'm showing you with all of the atomic detail that we simulate. And what we do is we calculate the forces and allow the forces to guide the motions by integrating equations of motion. How do we calculate the forces? Well, the atoms are simply point masses, but as you learn in a general chemistry class, a molecule is not just a collection of atoms, but they actually have bonds. These are bonded together, and so we need to include these bonds between atoms in our functions. And we also include angle bending because there are local geometries. Atoms take up space. We include van der Waals interactions. We have functions for electrostatics and salvation for this charged lysine, for example. And then there are other terms such as rotational barriers. So when we put these all together, we end up with a function that calculates the forces, and all of the parameters in this function are trained based on more complex physics such as quantum mechanics. We do this in a simulation program called AMBER. My group is one of the lead developers in the AMBER software, and it's used around the world for these types of calculations. There are many, many atoms in these systems. The ones I'm going to show you for the spike are over 1 million atoms, like this small virus. And if we run these simply on CPUs, then we get very short times, fractions of a nanosecond per day, which is on the order of some of the local motions, but not large-scale changes that we want to simulate. If we port our code to GPUs, which we've done for AMBER, we can get 20 to 50 nanoseconds per day, which is starting to reach to biological timescales. So these are expensive and require lots of GPUs. Are they accurate? One of the things we'd like to know is structure, and this is an example from my lab a few years ago, where we showed that using our energy functions in simulations, we could get proteins of various sizes to fold to very accurate structures compared to experiment, shown here red compared to blue. We'd also like to be able to calculate interaction energies and model those accurately, such as between the spike and antibodies, or ACE2. In this paper from a couple of years ago, we calculated the interaction energies between pairs of small proteins. So we have an equilibrium between the proteins apart and bound together, and we have experimental measures of how that binding energy changes as we make substitutions in the protein. And you can see that here the x-axis experiment, the y-axis calculation, they're in very good agreement. Finally, we'd like to be able to model motion, such as changes in the spike structure as it opens and closes. And about a decade ago, we showed for the important AIDS drug target HIV protease that we could simulate the opening and closing dynamics that allow things like drugs to bind to the interior of the protein. So even though we make a lot of approximations, we're doing a reasonable job. Here's our simulation system that I'm going to show you today. It's the spike with all of the atomic detail, and in green are shown the glycans, the sugars on the surface that also help it hide from the immune system. We have a few 100,000 water molecules, and we have salt. And overall, it's over 1 million atoms, and it's a droplet about 22 nanometers in diameter. So what do we learn from these simulations? I'm showing you now how we can map the pathway as this receptor binding domain opens and is able to bind to an ACE2, so it must open to search for it. And we see the formation of a transient pocket in a very highly conserved region that serves as a hinge about which the receptor binding domain rotates. If I zoom into the protein interior to give you a better view of that hinge, going past here the glycans and the rest of the protein shown as the surface, you can see this pocket and all of the atomic detail that we're simulating. There's a large space here when the receptor binding domain is open. And as it closes back down again, you can see that this space collapses and these two surfaces come into tight contact when the hinge is fully closed. We want to be able to map this and describe this in more detail, so I'm showing you just one of our 3 monomers, or identical proteins, with a series of time snapshots showing you the opening and closing motion. And we're going to map that using 2 descriptors. One is going to be an angle describing how open the receptor binding domain is, and the other is a rotation of that domain around the main spike axis. We can take those 2 coordinates and map grid points where we've seen structures during our opening motion in the simulations. Then we can deploy a large number of simulations to do local explorations of that grid. And that can be done in a cluster, in this case with 300 simulations. And that allows us to calculate a free energy landscape. The free energies are what control things like populations. And so low free energies, shown here in blue, are favorable and stable, and the lighter colors like yellow are higher energy states that are less populated. Consistent with the experiment, we see the lowest value here in our free energy landscape to be the closed spike structure, and less often it opens. We can ask whether the spike is sensitive to changes in this pocket since it's very highly conserved. And we see alanine at position 522 in the normal virus. It's exposed to water when the receptor binding domain is open, and when the hinge closes, it's tightly packed in the interior. We hypothesized that due to this tight packing, a larger amino acid would not be well tolerated and would prevent the spike from closing all the way. So we tried to increase the size of this amino acid by designing our own substitution. So this is our normal non-substituted spike with our free energy minimum in the closed form. We're going to replace alanine 522 with a leucine, which is 3 carbon atoms larger, not much larger. And you can see that even that small change has shifted our free energy landscape to where we now favor the open form. And although it can be closed, it's strained compared to the form before. So that's consistent with our hypothesis and our prediction. When we look at clinical strains of SARS-CoV-2, we do see instances of an A522V substitution. V is for valine. Valine is not as large as leucine, but it still seems to have an impact because it increases the antibody neutralization in experiments. That is explained by our landscape, which shows that larger amino acids in that position shift the spike to a more open position, which we know is vulnerable to antibody binding. We're still not sure why this would be seen in the virus in circulation, unless perhaps it compensates for another substitution that makes it more difficult to open the receptor binding domain, and this helps compensate. One problem with studying these and understanding them is that it's difficult to make and purify many of these variants that result in more flexible spikes. Experimentally, they don't purify well, and so that makes the simulations even more important. One possibility for helping experimentalists study the more open forms of the spike would be to design a small molecule that would serve the same purpose, and it would shift the equilibrium to a more open state. So we took our structure here for this state with the hinge open, and our colleague Rob Rizzo at Stony Brook carried out a virtual screening against a library of small molecules and found ones that fit into that pocket and made the appropriate contacts. When we do simulations of the spike, either without the small molecule or with the small molecule in the hinge pocket, shown here in pink, and compare them, we can see that in the absence of the small molecule, as we saw before, the spike does open and close, but it prefers the closed state. However, with the small molecule bound to the hinge pocket, the spike is unable to close due to the steric clash. The small molecule stays bound and the spike stays open. When we quantify those observations by calculating the energy landscapes, this is the same one as I showed you before for our normal virus, nothing bound. And now for the exact same spike protein, with the addition of the small molecule, it results in a much flatter free energy landscape and more exposure of the spike into an open form. But that can be done now with the addition of a small molecule after the purification of a protein and doesn't require changing the spike sequence. Now we're extending these calculations to be able to look at more complex systems, such as this one here, where in pink, I've added in the ACE2 receptor, and now a receptor binding domain of the spike is opening as it interacts with ACE2, and we can carry out the same kinds of calculations there to look at the impact of changes and binding of small molecules. We were fortunate to be part of a team that last year was awarded the Gordon Bell Special Prize for COVID-19, and one of our other team members, Lillian Chong, is also giving a talk at GTC, and I encourage you to see that talk if you're interested. I'd like to thank my team members. We're a very diverse team with a number of graduate students and a large number of undergraduates that help with research. I'd like to thank my collaborators, our funding sources, and especially the COVID-19 HPC Consortium for providing the large numbers of GPUs that we need. And if you're interested, you can ask me questions in the chat or check our website that's listed down below. And thank you for your attention.